Transcription begins in the nucleus of the cell. The information in DNA is transcribed into a different molecule called RNA. In our model, we only have one strand of DNA, but normally it is double-stranded. An enzyme called RNA polymerase unravels the DNA in the 3 to 5 direction, while pulling in nucleotides to make the RNA strand. This process begins at a promoter site on the DNA and stops at a terminator site. The new strand being created is called mRNA, or messenger RNA. RNA has the same base pairings as DNA, except uracil replaces thymine. The DNA strand that the mRNA copies is called the antisense strand. The strand of the DNA not being copied is called the sense strand.
In the DNA, there are coding parts of the gene as well as non-coding parts. The coding parts are called exons and the non-coding parts are called introns. Both parts are translated into mRNA. However, after being translated, the introns are spliced out of the mRNA. We show this in our model by coloring in the blank pieces of our mRNA strand. After the mRNA has been spliced, the new molecule exits the nucleus via nuclear pores.
Translation is protein synthesis, in which the first step is initiation. The mRNA travels into the cytoplasm where it teams up with a ribosome. The ribosomes build proteins by translating sequences of 3 nucleotides, codons, in the mRNA into a sequence of amino acids, which are the primary structure of proteins. Transfer RNA, tRNA, has anticodons that should match up to the codons within the mRNA strand. Anticodons are 3 nucleotides that match up with the nucleotides that make up the codons on the mRNA. The tRNA floats around in the cytoplasm, where it picks up the correct amino acid that its anticodon codes for.
The anticodon on the tRNA matches with the complementary mRNA codon in the A site of the ribosome. Then the connected mRNA and tRNA move to the P site, freeing up the A site for another tRNA to match its anticodon to a new mRNA codon. The amino acid attached to the tRNA in the P site detaches from the tRNA and forms a peptide bond with the amino acid on the tRNA in the A site. The mRNA shifts through the ribosome again, moving the attached tRNA with it. The tRNA that was just in the P site now detaches from the mRNA and goes bye-bye back to the cytoplasm to pick up another amino acid. The tRNA that was in the A site is now in the P site and is connected to the growing polypeptide. This is the elongation phase of translation.
The last step of translation is termination. This is when the stop codon on the mRNA is reached. There are 3 stop codons, UAA, UAG, and UGA, and it can be any of those. When the stop codon matches with its complementary anticodon of tRNA in the A site, the tRNA doesn't have an amino acid attached to it. The polypeptide attached to the tRNA in the P site detaches from the tRNA and is released into the cytoplasm, where it will build its secondary, tertiary, and possibly quaternary structure. Meanwhile, the tRNA is detached from the mRNA, and the ribosomal subunits break apart again.
That is the end of the transcription and translation process. Another thing to make note of is that the whole translation process takes less than a minute. In addition, multiple ribosomes can translate a single strand of mRNA at the same time. Once the start codon has passed through the first ribosome, another ribosome can attach to the mRNA and begin translating it as well.